All right, we're going to go ahead and get started this morning. Welcome to the first in our April Produce Safety webinar series. We have three really on-topic webinars scheduled for our audiences this month. Our audiences being our produce growers and anyone who works to support their work and is engaged in the produce industry. So we're very excited today to have a presentation from Dr. Waite-Cusic. She's the Associate Professor of Food Safety Systems at Oregon State University in their Department of Food Safety and Technology. I'm going to give her a brief introduction, and then I've got just a few slides to go through, and then we will let Dr. Waite-Cusic put up her slides and take it from there. So Dr. Waite-Cusic is an Associate Professor of Food Safety Systems, as I just said. She is also a co-PI for the Western Regional Center to Enhance Food Safety and a member of the Oregon Farm Food Safety Team. She oversees a large research program on the microbial food safety system food safety throughout the food system. And today she's going to highlight a few of her active research projects focused on food safety. We thought this was a great opportunity to have her all to ourselves over here on Idaho, even though some of our participants may be from Oregon and other parts of the country as well. But it's really going to, I hope, be applicable to many of your operations and your work. And it's just great to hear about research that's happening in our region. So just a few slides that I have to get us started. Again, most of you have probably been on webinars before, and this is not a brand new thing, but just to be most successful in your experience and have the best experience, it does help if you close other programs that are running on your computer. If your internet is a little bit bouncy, if you're having weird weather today, those are important things. And if you check your sound, the buttons on the Zoom, you can change microphones. Sometimes if you're having issues, you can also call into the webinar using the email that you received, and you can listen in on a phone if you have to. We don't have an open, we can't see you today, but if you do have questions, we are going to be actively responding to them. So you may use chat or the Q&A feature to put your questions in, and we will pause if we get one that we absolutely have to answer during the webinar. If not, we will save them till the end. If we have any handouts, we will send them in a follow-up email. We don't have any handouts for this webinar ahead of time. And just this was something we needed to put in, that when we're talking about produce safety and food safety, we're not talking about COVID-related problems or questions in this webinar. So our goals for today's webinar is that we really hope that you will learn more about some relevant research that's happening related to produce safety, increase your knowledge of the food safety risks that are preempting this research, and be more prepared to implement best practices in your operation or in your outreach work. And when we come to the end, I have a couple more slides that talk about what's coming next, a little bit of follow-up, and there will be a survey. But that's all I had to start today's webinar. So Dr. Waite-Cusic, if you would like to share your slides, and then I will turn the controls over to you.
Great. All right, can you see them?
Yeah, that looks wonderful. Thank you.
Excellent. Well, thanks for having me. Today, I'm going to try my darndest to give you an overview of 3 different projects we're working on. So it's a little bit much, and they interface in unique ways each day as we move forward. So this is certainly not just me that's doing this work. So I have a huge group of collaborating PIs here at OSU and at different institutions. And then my team makes this all happen. So you'll see some friendly faces as we move along, but I'll jump in, and hopefully this will be somewhat entertaining. So we creatively call ourselves the OSU Food Safety Squad, and we are a large team, and I will say the boundaries of that team are a little bit blurred at times. So this is the majority of my graduate students and FRAs. And then in the middle is my PhD advisor, who came to visit a couple of weeks ago. So we were really thrilled to have him here and see what's come of his training of me. So it takes a village, and he was a very important part of that. As a team, we are the Food Safety and Quality Systems Lab. So that's my group specifically. And we're really here to serve as a resource for the food industry. And that extends to farms and also extends to consumers at times. And we're really here to try and understand any sort of microbial issue that they're facing in the real world. I like to joke that I'm not a creative researcher. I would much rather deal with problems that already exist than to come up with problems of my own. So I do wear an outreach hat and quote unquote extension. I say that quote unquote because I don't formally have an extension appointment, even though I act in that capacity on most days. So there's a lot of things that we provide. Often it might be just an email or a phone call to help answer a question. And then that will extend to process validation studies or short-term research projects for the industry. And then we also do the sort of typical long-term research. And I'm going to focus on that in this presentation. But we've worked with hundreds of companies over the last 12 years, I guess. It's almost been since I've been here. But we're really trying to be that help so the industry can understand what's going on and hopefully help them find some solutions. So we're here today talking really about produce safety and microbial issues related to produce safety. So all of you on this call are very aware of the produce safety rule, I'm sure. But I think this slide really helps us put some context around everything. So this is how at least, FDA buckets the risks of produce contamination. And then thinking about how to implement those on your farm to reduce the risk of microbial contamination and foodborne illness. So, the typical ones, right? Biological soil amendments of animal origin, worker health and hygiene, wildlife and domestic animals, ag water, and post-harvest handling and sanitation. And we're in full implementation here, right? Other than ag water, which I apparently picked the worst category to focus my own efforts on, which was ag water. And so I feel like I'm never going to get out of it. But it's been a lot of fun to learn more about the water system and water use on farms. So you'll see that sprinkled in as we move forward. So I wish I had $1 for every time I heard an industry member say this to me over the years, right? There's never been an outbreak in, and then insert whatever product, food, whatever it is, right? And I say this a little bit tongue in cheek because I have heard it so many times. And then unfortunately, right, we'll turn around and there is an outbreak with that particular commodity. I think this is starting to go away, right? Because enough industries have sort of been bitten in the butt by this, but we have to create this food safety culture and continue to train in this area so that people understand challenges with their system. So this is sort of the response to that statement, right? Of we have had so many outbreaks and so many different types of fresh produce, and estimates are about 30 produce related outbreaks a year, depending on where you put the cutoff in size. This often leads to 18,000 confirmed cases, which means we're dealing with many more that are not confirmed. But you can see the diversity here, and these all grow in a diversity of growing regions and harvest and post-harvest conditions, and on and on. And so trying to mitigate that risk as best as possible is a challenge for sure. So with that context in mind, I'm going to share 3 ongoing research projects that we have in my lab. And again, some of these are definitely in collaboration. So I am leading a Center for Produce Safety grant that's looking at risks related to pre-harvest water applications in dry bulb onion production. I'm also involved in a USDA CARE grant. This is led by Ivana Kovacevic at the Food Innovation Center in Portland. And we're looking at cleaning and sanitizing optimization, basically for farm use. So we're talking about post-harvest handling and sanitation for that grant. And then we have a new grant from the Washington Specialty Crop Block Grant looking at dry cleaning in onion facilities. So mostly talking about post-harvest and in the packing house, and what we can do there to help minimize at least cross-contamination. So we'll talk about each of these in detail with kind of an update of what's going on. So we'll jump in with Center for Produce Safety. So I've worked with the onion industry for a long time, mostly when the produce safety rule in the ag water section first came out. And we were really dealing with this approach of it's never happened to us, right? Like we've never caused an outbreak. And so that was the goal of our initial research, was to prove basically the production practices that help to minimize risk. And we did that work in 2015 about. And then 2020, along with COVID, right, we also dealt with this giant salmonella outbreak that was linked to red onions grown in California. And this was the largest salmonella outbreak that we'd had in about a decade. So this was a big deal. And we were dealing with this response, and it was cases in the US and Canada. So this was a big deal. FDA and CFDA, or CDFA, I think is actually the acronym, did the outbreak investigation for this particular one. So they both were on site in California to the farm that was the onions were traced to. And there were actually 2 growing regions that were associated with the outbreak cases on the trace back. And they collected a lot of samples, environmental samples. But you can imagine that this was not during the growing season, like it had been a period of time after active growing. And so the conditions aren't exactly the same as they were at the time of harvest, but they did their due diligence to collect lots of samples and to analyze those, particularly looking for the outbreak strain. From this evaluation, they did take over 1,100 onion samples from the packing house, and none of these tested positive for salmonella. They did collect over 800 different environmental samples. 26 of those were positive for salmonella. 11 were positive for salmonella Newport, which was the outbreak serotype, but none of these isolates were the outbreak strain. So we don't have any additional data from this sampling effort to really point to a potential source for this particular outbreak strain. We have some hints maybe from the detection of other salmonella in the environment, but nothing that's really clear. So when you can't find the outbreak strain, you tend to speculate, right? Because we need to answer some questions and try and give industry and the regulators some direction of what may have happened. The problem is this leads to speculation because we really don't have any additional data that supports it. So we're really pointing back to things that we've been told through the produce safety rule and through other outbreak investigations. So the leading hypothesis from this report was really contaminated irrigation water, but then there were lots of points to other things, again, that underlie the produce safety rule. So adjacent nearby land use, the presence of livestock, there was other plausible potential sources of irrigation water, or sheep on adjacent land, or animal intrusion, or lots of birds, right? The packing house conditions were not particularly sanitary, which is pretty normal for this industry. And then there was pest and animal intrusion in those packing houses that was obvious. So all these things I've highlighted on the right, are the only thing they didn't point out was worker health and hygiene, right? So otherwise it was, everything else was fair game. Okay, so that one kind of blew over, and then 2022, the same thing happened. So we see another salmonella outbreak, again, a huge one, over 1,000 confirmed cases, and these were imported onions, again, red onions, coming from Mexico. So we have these 2 outbreaks associated with a commodity that really had never had an outbreak before, at least in whole bulk onions. And so what can we learn from analyzing these isolates and other things? So FDA did do their normal whole genome sequencing and analysis on these strains, and there were some interesting things about both of these outbreaks. In both cases, all of those clinical isolates within an outbreak, so within the 2020 outbreak and within the 2021 outbreak, all those clinical isolates were very, very similar, more similar than we see in a lot of other outbreaks. So only 0 to 2 or 0 to 4 nucleotides difference. So within the whole genome, there were only up to 4 bases that differed, despite going through an agricultural system and through a human system and through all the distribution that went in between. So this is really, to me, a very interesting situation. And so one of the questions that I've had is, how do you really get a population this similar, this high and this similar, that infected so many people? And in this case, the only conclusion I can come to is that there was one particular strain of Salmonella that had to be growing somewhere in the system to a very high cell density, likely by itself, because it didn't have any other genetic inputs or manipulation. And then it contaminated a lot of onions and then survived during storage and distribution. So that's the hypothesis I'm working on, or the underlying assumption that I'm working on as we design some of these studies. So let's jump into the CPS project in more detail. So in this case, we were really looking at the application of contaminated water late in the production season, so before harvest, because they don't add any water after. So we're really looking at that risk right before harvest. And from our discussions with industry and with our onion extension specialists, we identified a couple of water applications that were very late in the season. So those were the ones we wanted to focus on. 2 of those applications were related to pesticide or crop protection applications using water. So one was a pesticide application, and then the other was the use of clay to protect from sunburn. So in this case, there is a potential that those chemicals or the clay could be mixed with water that's contaminated with E. coli or Salmonella, and then the application of that water onto the crop could lead to contamination. So that's how we were looking at that potential risk. We evaluated that in Oregon last year in Malheur County, in Ontario at the Malheur County Experiment Station, and we looked at white and red onions in that trial. And so we did a pesticide application with contaminated water and a clay application with contaminated water, and then sampled those onions during the field curing period before harvest. Similarly, in Washington State, we were more interested in overhead irrigation as a risk. So we compared overhead irrigation with contaminated water to drip irrigation with contaminated water, and we focused in this case on yellow onions. Didn't really matter. There was just a question from the grant to look at the potential for different varieties to behave differently. So that's kind of how we split this off. Again, we were looking at the survival of E. coli from that last irrigation event with contaminated water and then through the field curing. So if you've never done a field trial, it is a logistical nightmare, especially when you're dealing with supply chain issues that happened and continue to happen these days. So this is just an example of some of the things we were dealing with. Our field trial sites were not down the street. Okay, so we were dealing with managing a field trial in Pasco, and we were dealing with managing a field trial in Ontario. And we were the microbiologists. Our field extension specialist did an amazing job managing the field and making sure the crops were healthy by the time we got there, but the micro was on us. So we had to pack up and take all our stuff, clear across the state in order to be able to accomplish this work. So it was a definite undertaking. I don't know what I was thinking when I proposed this, but we did it. So again, let's look at this Oregon field trial. So again, we're really focused on pesticide or clay applications when those are prepared with contaminated water. So on August 23rd, we did that that contamination event and then monitored it over time. So these are just, I'll show you some pictures of the field because I think it's important to see sort of what we were dealing with. In this case, we had a white variety Cometa and a red variety Red Wing that were grown on site at the experiment station. And we had 4 plots per each variety that were treated with pesticide, and then same for clay. And you can see the sort of relative maturity of the plants were quite different at the day of inoculation. So the red onions had really struggled last season due to the heat. They're on the right. And then on the left, you can see the white onions were doing quite a bit better at the time of irrigation. But regardless, this is what we were dealing with when the time came. So this is kind of what it looked like as we did the contamination event. So you can see in the hazmat suit, right? This is our pesticide applicator, Ian. And I contaminated the water for him. He mixed the pesticides and then away he went with the backpack sprayer to contaminate those fields for us. And then you can see my team in the PPE that's walking behind him to collect the samples. Oh, we don't need to watch the video of him walking. Okay, so again, this was us. We decided on inoculation day to collect samples at time 0, so immediately after the contamination event, and then again at the 6-hour mark. So we were back out in the field in the heat of the day collecting these samples, and the students are still grumbling about how much they were sweating. I should note that I had ruptured a tendon in my foot 2 weeks before this field trial, so I was walking in a boot at this point. So I couldn't collect samples, but I was very much active in the lab on those things. So you see me standing here, I'm a little bit less than normal functionality. So if you haven't seen clay application, I think it's kind of important to see how that's distributed on the plant. And I'm not saying that this is perfect. I think if you were using a tractor applicator, I think the clay distribution would be a little bit more uniform. But this is really what we're looking at. It is splattering the clay onto the surface of the onion. And it would certainly also, it gets all over the leaves, right? So there's just this smattered application of clay and potential contaminant. And it definitely dries onto the surface of the onion. And we believe, right, the clay itself is protecting the onion from UV damage from the sun. So we also think it's probably going to protect bacteria that may be either mixed in with that clay or remaining on the surface of the onion. So those are kind of the working assumptions for that. We also collected samples at the 24-hour mark. So this is just us back out in the field. It's always a beautiful experience to be out in Ontario and doing that work. And then I mentioned that we were off-site to do the initial collection because we couldn't actually get samples back to the lab fast enough with the 8-hour travel time. So we set up a lab in the Malheur County Experiment Station. Stuart did a great job facilitating that, and we had plenty of room to do our work. So you can kind of see the 2 stations where we were setting up our samples. So let's look at the results from this particular study. So in this case, we've sprayed onions either with contaminated pesticide or with contaminated clay, and then we've collected samples from each replicate plot to figure out how many of those onions were contaminated and at what level. So keep in mind, we expected to see a pretty significant loss or lack of recovery from the actual onions because a lot of that contamination event is going to be overspray. And also we're going to, the bulb itself is going to be protected because a lot of the application is going to be applied to the leaves. So these are the results related to how much E. coli we applied to the actual bulb, like a representative area. So this is basically what we're expecting to apply. And then, so 7.5 to 344 cells per onion, basically for the pesticide applications, and then a similar amount for the clay applications down here. So that was the water actually sprayed out of the system that hit that surface area, that estimated bulb. And then when we collected the samples, we collected 10 per plot, and then we tested those for the presence of the E. coli that we applied as a surrogate for salmonella. So in this case, you can see that the majority of the onions were actually contaminated by this process. So 9 out of 10, 10 out of 10, 7 out of 10, 9 out of 10. You can see that through the whole data set. And then this is the approximate number of cells that were recovered from any particular onion. So this is an average. So we're dealing with about 7 cells per onion, 18 cells per onion, 3, 10, etc. So you can see that this application event led to a majority of the onions being contaminated, but certainly not all of them. And then the level of actual contamination on the bulb was quite low. So on average, somewhere between 3 and 10 cells. And this was using water contamination levels that were representative of the region and the risk that we might expect. So this is the starting point, time 0. So then as we monitored those same plots over time, we collected samples again at hour 6, and then day 1, day 2, day 7, day 16, and day 29. And what you'll see is this decrease. So from day 0, right, we had the majority contaminated. At hour 6, we've already seen a pretty substantial decline in the ability to detect E. coli in these bulbs. That continues to go down after 24 hours. And then from day 2 on, we only ever detected E. coli on one bulb at the very end of the season. So this field curing, right, is very rapidly leading to a decline of E. coli that were applied using this method. So we don't know about this, 1 out of 80, is that representative? There's a lot of questions, and it's obviously very difficult and takes a new strategy to investigate very low occurrence, very rare problems. So we're definitely evaluating that strategy as we move into our second year here. But regardless, the pesticide in the clay, we really saw huge reductions between application and after day 1, and seemingly very limited risk to this type of water application. As we move to the 2022 field trial, in this case, we're looking at overhead drip irrigation, in this case on yellow onions. So similar thing, we were at a beautiful field site in Pasco, and we utilized the drip tape that they laid for us to inoculate those plots, and then we used a tractor equipped with a spray bar to do the overhead irrigation for this particular trial. You can see that the onions in this case were very healthy. They were nearing the end, but they were certainly more healthy than the ones in Oregon at the same time frame. We chose to irrigate basically as high as possible for this particular event to sort of maximize the potential for contamination. Excuse me. So we targeted 0.3 acre inch, which is a high end for Washington, but reasonable. And then we hit that same level of irrigation using the overhead application. I do have a little video here. I'm going to apologize if it's loud, but just so you can see how this works. So we added E. coli to this tank, and then they drove the tractor down the plot in order to contaminate the onions in a realistic manner for overhead application. It's not perfect, right? Because, it's not a center pivot. It's not the typical physics, but it gave us sort of the maximum opportunity for contamination. Oh. Okay, so in this case, we were hosted for our lab at the Franklin County Extension Office there in Pasco. You can see me trying to get down the stairs with my crutches. I just went down on my butt instead because I have no shame. And so you can also see our little lab setup, and it was a great experience working there as well. So at time 0, we're going to compare drip irrigation and overhead irrigation. Okay, so in drip irrigation, immediately after the end of our irrigation event, we're seeing none of the onions positive for E. coli, for the contaminant. We kind of expected that. Okay, so it will take time for any contaminant in the water to move through the soil to eventually touch the onion and be detectable. So at time 0, no detection. But with our overhead irrigation, we applied 907 cells approximately per onion bulb's surface area. And then what did we actually detect on the bulb? Well, the majority of onions were positive, and our rates ended up being around 10 cells per onion bulb. This is really because the tops, right, you saw how healthy they were, really shielded the bulb itself from becoming contaminated from that water application. So as we monitored those onions over time through field curing, we saw some interesting things. So on the drip irrigation, we had day 0, none of them were contaminated. And then at day 1, as that E. coli moved through the soil, we did see some positives. So we had 5 out of 40 onions were positive at that point, with an average load of about 7 cells. And then that was it. We never detected E. coli again on those onions that were contaminated with irrigation water. This is real comparable to studies we've done in the past in Malheur and in California. So this is just confirming drip irrigation poses very little risk here. When we go to overhead irrigation, we've got a different story. So we've got at day 0, right, the majority of onions, almost all of them were positive. Day 1, we start to see a decline in the prevalence. Day 2, right, this continued decline over time. But eventually at day 28, we aren't detecting anything still. So field curing definitely does seem to be a risk reduction strategy, even in higher levels of contamination. I do want to point out this particular onion. So on day 15, we did have 1 positive onion, and that onion actually had a higher load than what we expected. So we have some questions about that. Is it an outlier or is it real, right? Is it a real situation? So I haven't told you how many onions that we examined through this whole study, but it was 2,000 onions that we did through the 2 field trials. And my poor students, I forced them to take photos of each onion and to weigh all of them and do all this stuff because we weren't sure when we would have something like this that we needed to investigate. So this is that onion. Okay, this is the photograph of that one onion that had the high E. coli count at day 15. There's a lot of caveats to this, right? So we don't want to read too much into this one little onion, but this onion did have a problem. Okay, so it did have this internal scale, dry scale, but we don't know if that matters. Okay, also keep in mind, we didn't actually analyze the interior of these onions for this analysis. We really only analyzed the exterior of the onion for E. coli. But there are questions about whether as the neck dries and the syneresis happens on the neck tissue, could E. coli grow in that space or even down in this early area of the neck as the scales close? Is that a potential option? So we don't know. We really don't have an answer. We're trying our darndest in the lab to figure that out and test these hypotheses. But this, I do have to use at least one of the photos of the millions that they took. So this is a good one to highlight. So as far as the field trials go, what have we learned so far? Well, last year, the late season clay and pesticide application really created a low rate of contamination, and that died off very quickly. Likely due to the really fast evaporation rate of a very low volume of water that gets applied. Overhead irrigation does create this higher risk of contamination. I didn't finish that sentence, I'm sorry. But the surface contamination may or may not correlate with foodborne illness risk, right? So if we have contamination on the surface of the onion, that doesn't necessarily mean somebody's going to eat it, right? We peel it. There's concerns about transfer as we cut the onions, but we really don't know. And we've never looked at internalization before. We know that there's issues with internalization and the potential for the internalization of bacteria from plant pathology studies. So it's quite possible. They tend to correlate those rots with late season water applications. So we're seeing some correlations and we're asking a lot of questions about this. So this is an ongoing area that we're studying as we move forward with the onion work. So we're planning on the 2023 field trials for that grant right now. And I think the onions are being planted this week. So send us positive thoughts come August when we'll be out in the field again. Okay, so we're going to move on to the USDA CARE grant, and I'm not going to have as many details on the next two because they're a little bit more straightforward. But the USDA CARE grant is really focused on post-harvest cleaning and sanitation. And we're really dealing with this challenge, or the industry's challenge, related to the particular portion of the regulation and all its vagueness. So I'm sure you're all familiar with, but I wanted to throw this up here, is we're dealing with the expected compliance of you must inspect, maintain, and clean, and when necessary and appropriate, sanitize all food contact surfaces of equipment and tools used in covered activities as frequently as reasonably necessary to protect against contamination of covered produce. Okay. When necessary, who decides that, right? As frequently as reasonably necessary. Who decides that, right? So we've got two of those in this one regulation. And then we've got this expectation to clean and then as appropriate, sanitize. Who decides what appropriate means, right? So just as a review, again, you guys all know this, but to get everybody on the same page, what are the 4 basic steps of cleaning and sanitation? And we're really dealing with cleaning having multiple steps. So a physical removal of major debris from the surface, right? This might be brushing or blowing or something like that. And then the application of an appropriate detergent, right? Or some other sort of emulsifier to be able to break down some of the more harder to remove debris and organic material, and then rinsing that surface with clean water. So those would be the cleaning steps. And then sanitizing is an additional separate step after cleaning. And this would be the application of a sanitizer on an already clean surface in order to reduce the presence of microorganisms. And this is going to require that you use a sanitizer that's approved by EPA, right? And that requires that you follow the label because that's a federal law. And so those are things that the industry, I think, is still, in some cases, still grasping sort of the necessity of following the label as prescribed. So we had a lot of questions about what farms are doing related to cleaning and sanitation. And so we conducted a nationwide survey. It ended up being sort of hyper-focused in the Pacific Northwest because that's who responds to our surveys. But we wanted to get this idea of like, are people rinsing only? Are they cleaning only? Are they doing cleaning and sanitation? Or are they not doing anything? And for what sort of category of food contact surfaces that might be touching their covered crops. So we bucketed those in different categories: transportation equipment, harvest equipment, handheld tools, harvest containers, storage containers, and other post-harvest equipment. And you can see kind of the frequency of the breakdown here. We were most interested, right, in what was going on with cleaning and sanitation, but we definitely had interest in, you know, what's going on with just rinsing or with just cleaning. And then if they were using a sanitizer, what type of sanitizer were they using? And so we asked a follow-up question for that. So the majority were using bleach, a lot were using quats, some were using peracetic acid, and then some were using other types of sanitizers. You'll see that these percentages add up to more than 100 because many operations use more than one sanitizer for different reasons. So from this, we wanted to start to evaluate in the lab how effective cleaning was and how effective sanitation might be from a well-controlled system, right, on a surface. So in this case, we decided to first work with stainless steel as it's common in the industry, and it's also very easy to work with, right? It fits all the rules. So it's a non-porous surface, it's definitely used, and so we want to know what would happen. So we contaminated that surface with listeria in the presence of some organic material. And so you'll see on the far left here, this is our dirty surface. And so this is very contaminated, right? So over 8 logs per coupon. And then as we applied different steps in cleaning and sanitation, we wanted to monitor that reduction. So if we simply rinsed that with water, we got some reduction. So we got almost a 2-log reduction from just rinsing, which is good news. The whole cleaning, so a dry step, a clean step, and a rinse step, really didn't do much more than the rinse step in this case. And that's probably because in the stainless, there's not a lot of opportunity for really tight bonds on the surface. And so they're probably pretty easily removed. We're seeing that. And then if we dry clean with a detergent, rinse, and then sanitize, so cleaning and sanitizing in the right way, then we see additional reduction. Okay, so this is with bleach. this is with peracetic acid, and this is with a quat. In this case, we're definitely seeing improvement over just cleaning. And what's interesting is we wanted to evaluate a drying step as well, because a lot of facilities will do their thing and then let things dry. And so for peracetic acid, that was really important. So we went from, some level of reduction. And then if we added a drying step, we had no detection of Listeria after that drying step was over. So that was really important. We also wanted to evaluate sanitizing the wrong way. So meaning I just applied a sanitizer. I haven't done what I was supposed to do and cleaned first. And we wanted to know if that would have any sort of influence on whether or not these surfaces were cleaned. This is not in alignment with the label, right? The label says that you must clean first and then sanitize. That's what the regulators are expecting too. But we did want to understand that. And there are some products on the market that are mixtures of cleaners and sanitizers. And that's an area that we need to do some additional training on, because there are different instructions for those products. But same story, we did see sanitizing by itself did work. And it was better than cleaning by itself, but it was not as good as sanitizing the right way. However, what happens on the stainless steel doesn't necessarily happen on every surface type. Okay, so sanitizers are designed to be effective on non-porous surfaces like stainless steel or plastic. In this case, there's been a lot of questions from industry on how to clean and sanitize wood. So in this case, we decided to go ahead and look at the efficacy of these same practices on plywood. So we were thinking about these like harvest containers, harvest bins, that kind of stuff. So you can see that, again, a wet cleaning provided very minimal loss of Listeria in this case. And then if we added the full dry cleaning, sanitizing, the whole shebang correctly, we did see some reduction, certainly not as good as stainless steel, which is what we expected. But we did see good reduction, and in this case, PAA seemed to perform a little bit better than the other two, especially with drying. Sanitizing the wrong way was pretty comparable to both cleaning, and it was right in the middle between cleaning only and cleaning and sanitizing the right way. So anyway, just some interesting data that we're going to be able to use as we move forward with this work. We also had questions about, so this was all in the lab under very tightly controlled circumstances, and we were using Listeria monocytogenes as the target organism. But what happens in the real world? And we had questions about how those cleaning and sanitation practices might be applied in the real world, and whether we could demonstrate their efficacy there. So we were able to partner with actually 5 different commercial processors to do this study. I'm only going to present the results from a couple of these today, but we picked to present here the ones that were the most different. Okay, so we worked with a facility that did, they were post-harvest packing house, mostly for frozen blueberries. And they would intake these green plastic bins. And then after the fruit was dumped for processing, then these bins would go through a bin washer. Okay, and this bin washer had a detergent applied, and that was it. So cold water, detergent, automated system. Okay, so wet clean only. Then we worked with a blueberry harvest operation that did hand harvest with buckets. That one did a wet clean and sanitation in your, I'm going to call it your typical commercial kitchen type situation. So a near perfect cleaning and sanitation system of a 3-basin sink and, the whole deal. So that was sort of our best case scenario here. And then we also went to an onion packing house that does dry clean only. And I'll present that data a little bit later as we get to the last grant. So wet cleaning did not reduce the microbial load on plastic bins. Okay, so we ran these dirty bins through the bin washer, and there really was no statistically significant difference between the dirty bins and the clean bins when we looked at aerobic plate count. So just typical bacteria. There was a little reduction, but it wasn't consistent. So there was a lot of variability here. But regardless, these levels are not super high. So they're managing the contamination level. And I'm sorry if you hear construction going on in the background. They're remodeling outside my office. Okay, so then when we look at the blueberry bucket operation that had a wet, clean, and sanitation, the near perfect system, right? Clean with a detergent, rinse, and then sanitize with bleach. We saw exceptionally good reduction in microbial load. So we were starting at, north of 3.5 logs per centimeter squared. And then after our 3-step process, we were getting very low levels of contamination. So almost a perfect inactivation here for removal. Okay, so we've demonstrated, right, some things work in real life and some things don't work in real life. And we're doing the analysis to see how the lab data compares to the actual in production data. But we also have some questions about the frequency and whether that matters. So how often are firms cleaning and sanitizing? And you'll see it's kind of all over the place, right? So we had some places cleaning daily, some weekly, some monthly, some seasonally. And then we have this bucket of as needed, right? Which is in compliance with the terminology and the rule, but doesn't provide us any real information on what that timing might mean. You can see as we get closer to what I'm going to call a consumer, right, or a processing environment, we're seeing typical daily type of sanitation management, which is what we expected. But there's certainly some spread in this data. So I wanted to share, because I think this is really important, to think about how surfaces look when they're in production and how dirty they can become, quote unquote, dirty or contaminated, just with normal production activities and during a normal production window. So if there is sufficient moisture and food and time, then bacteria can attach and grow quickly on food contact surfaces. So these are examples from another study we did looking at the attachment and growth of just native microflora on polypropylene and stainless steel. And the production shift we were looking at was an 18-hour production shift here. So this was what a polypropylene surface might look like. So you can see it's not perfect, right? It's got defects in it. It's got scratches and other things. Same with the stainless steel surface. Even though it's polished, it's not perfect. And the bacteria can like those grooves and nooks and crannies. And then as those surfaces are used more and more, right, more and more deterioration of the surface quality happens. So you can see after 15 hours of production, we're starting to see some attachment and even some growth, which is what happened here, where all these colonies look or cells look the same, meaning they probably replicated right there. And then with more time, right, we're seeing more attachment of organic material, which is all this crud out here. And then we're getting these nice little micro colonies where the bacteria have attached and are replicating. Stainless does a little bit better as far as not, like, they're a little bit harder to attach to the surface, and then it will take a little bit more longer typically for them to grow. A lot of that just depends on the nutrients and the sort of the quality of that stainless steel. But I just thought these are helpful to kind of see what goes on during a production day. Also, it's important to remind ourselves these surfaces are definitely not perfect. So you can see that sometimes there are grooves or cuts in different types of surfaces. These are polypropylene. So here's a cut and here's a ridge. And the bacteria can definitely get themselves sort of hung up on there and then start to replicate if they have food. This is just a blown up of this particular gap in the polypropylene, and you'll see, right, the bacteria have made a nice little home there, and it looks like they're also feeding on something that's in there. So they like to be in those protected places. As they get into those nooks and crannies, they're also protected from cleaning and sanitation. And so this is actually a belt from a food production facility, and you can see there's this kind of gouge in this belt. And as we zoom in, we can see that there's this nice little colony that exists here. And this was a belt taken after cleaning and sanitation. So this was what was still there, even despite a really robust cleaning and sanitation of a large piece of equipment. So maintenance and replacement schedules are really important to help manage these problems. I also think it's really important to remind everybody that cleaning and sanitation is not intended to produce something that's sterile. Okay, so we are trying to reduce risk, but we're never going to eliminate it, and it's not designed to do so. So I'll jump into the last one. This one is a new project, so we'll be quicker on this one. But this is really looking at post-harvest handling and sanitation in onion packing houses. So from the 2020 salmonella outbreak, we were really dealing with FDA trying to respond to an outbreak in a dry processing environment and how to deal with deciding where the lines were on what other product might have been cross-contaminated. So in that particular outbreak, the epidemiological link was to the red onions, but the recall got expanded to include yellow and white and sweet onions that had also been on the same packing line in the same facility. And that's because there really isn't a good clean break, as FDA defines it, in these facilities. And so how is the industry supposed to deal with this and defend what they're doing so that they don't end up recalling the entire crop? So this is the question we're really trying to get after in this particular research, is what is a clean break in a dry facility, and trying to understand that risk of contamination in those environments. So this is what we're dealing with. We have a contaminated onion go through and touch the surface, and then it leaves some bacteria behind, and then we have a new batch come through, and is it maybe picking up some onions? So that's just the context that we're trying to frame as we're moving forward with this research. And this is still pretty early in the investigation here. So we have these questions that we're trying to approach. So are the current practices effective? What are barriers faced by the industry to implementing some better cleaning and sanitation practices? And then what might actually work, right? So demonstrating and validating some of these. So I mentioned that we worked with an onion packing facility for that previous study. And so I'm sharing the data from that here. So onion facilities are dry cleaning. They usually employ some sort of sweeping, vacuuming, brushing, that kind of situation. And I've witnessed some of those, so I know firsthand kind of what it looks like. But dry cleaning, as we might expect, does not reduce microbial load on food contact surfaces in onion packing houses. And so we have data that really demonstrates that that's still true. So these are operations that are dealing with lots of dry soil, right? And just onions moving across those surfaces. And so we might expect those to be fairly contaminated. But we really don't want to get them wet, okay? Because once we add the moisture, now things can grow. Whereas most of the time, this is very dry and probably not going to support the growth of anything. We're just dealing with managing potential contact risk. And we're really fortunate that this crop has that outer skin. So even if we do have that cross-contamination, it's probably not a substantial risk to the consumer. But we don't know that yet. We have to measure it with some data. So we are looking for input from the industry, and we're doing quite a few interviews. So my graduate student, Sasha Nerny, is leading this charge. And we're using these interviews to collect a bunch of different information from onion packing facilities, really across the nation, but hyper-focused on in the Western region. So we're asking questions about current cleaning and sanitation practices, what types of food contact surfaces we have, what products have you considered for cleaning and sanitation, and what are some barriers to implementing those? And then we would like to do some more site visits so that we can observe what's going on already. So if you have anybody that wants to participate in this, or if you want to participate in it, feel free to email myself or Sasha, and we can make that happen. So as part of the grant, we are going to evaluate some sanitation procedures, and we're really approaching this from a practical standpoint. We don't want to reinvent the wheel. We also don't want to imply that these facilities will ever be as clean as an onion ring facility, right? That's subject to preventive controls. We're still very much looking at this as subject to the produce safety rule and how to manage that risk effectively. So we do want to stick with evaluating dry sanitizers. There are some traditional chemical sanitizers on the market for this type of application, and these are predominantly alcohol, but there are some that are combinations. So we are analyzing a combination alcohol and quat chemical to see how that behaves. There are also novel technologies that we want to investigate. So we've looked at UVC in the past. We want to apply that to some of the food contact surfaces here. UVC is challenged by shadowing, right? So if you have a lot of particulates, then it's not going to disinfect what's under the particulate, right? That's going to be protected. But even if you could sanitize whatever's on the surface, that probably would be helpful, right? So anything could help in this case. We're also looking at ionized hydrogen peroxide. We're actually getting a piece of equipment partially donated, and it should arrive this week. We're really excited about that, and the industry's been super supportive of that. And then ozone and superheated steam are two other technologies that we may look at in collaboration with Washington State. We're working on a grant application through the Organic Research and Extension Initiative program from USDA. That grant should go in, well, in the next 2 weeks. So we'll see if we get lucky with that funding, but really excited about these opportunities. So just going to close this out with some acknowledgements. I have an amazing group of people that I work with that are so supportive. The industry's been amazing to work with as well. And we just want to hear whatever we can from industry about their challenges or what's working or what data they may need to support their food safety efforts. Obviously, I want to thank my particular team. None of this could happen without them and our funding agencies. So, and thank you guys for having me today. I'm just going to close with, if you want to do an interview or send that information along, we can do that. But that's all I have.
Well, thank you so much. And I want to apologize, Joy. I have been reversing your last name.
That's okay.
I will change it on the slides. It's Waite-Cusic, not Cusic-Waite. And this was wonderful. One of the reasons that I really wanted you to be part of our webinar series is I find your presentations of research so engaging and friendly and easy to access. And I've sat through a lot of research presentations over my career. And so I'm hoping that our audiences appreciate that, that as well. Really good stuff. And I'm going to leave this up for just a second so that you can get her contact information if you're looking for it. And then I'm going to switch to my closing slides. But we do have one question in the Q&A right now. That's, can you share the names of the products used to sanitize wood in that study?
Yeah, this one is a little bit tricky. So I will have to pull the exact products. One of them was just bleach, right? So household bleach with a sanitation statement on it, right? So definitely an EPA registered product. And then we had a peracetic acid product and a quaternary ammonia compound product. So I can get the information for you, but I also need to be careful because the instructions for use, right, is that these would be applied to non-porous food contact surfaces, which wood is not one of those. So there is this, issue of you're not really in compliance with the labeled use. And so that isn't in alignment with all these things. I've been in a lot of packing houses, and some will actually, it was a pear facility I was in, they use wood bins and they'll actually dunk the bin into their produce water, right? So it's like they're dumping the pears into a solution that contains bleach, right, to keep the microbial load down. And so it's sort of inadvertently sanitizing the bin. So like there's quote unquote, I hate to say this, ways around it, but there are ways to manage the risk here that might be useful, that are still legal, right? You have to be careful with that terminology.
Thank you. I've got one more question in the Q&A. Have you published the results of the drip irrigation versus overhead irrigation study yet?
No, that was just this last year, and we're doing a second. We aren't going to compare drip this year, but it will be embedded in the same publication. We do have 2 other publications that have already been published for looking at drip irrigation. They didn't necessarily compare to overhead. One kind of did, the other one did not. So there is a paper that, well, I'm a co-author. I'm a senior author on one and a co-author on the other. So I could share those with Ariel if you'd like those.
That would be great. Thank you. All right, I did want to say that this is a great presentation to kick off our series because our series are based on input that we get from our growers throughout the year, input we get on our Produce Safety Alliance grower trainings when we ask folks what they want more information about related to produce safety and the FSMA produce safety rule. And then also it's based on the observations that our inspectors make when they're out in the field. And this past year, while I don't get the details of those inspections, I don't get any identification of where this came from, but overall, the primary observations had to do with water use and pre and post-harvest sanitation, cleaning, and et cetera. And so this is really where we're focused this year on our webinar series. So this one that you're viewing now or watching the recording of is the kickoff. We have another one on April 18th, which is really focused on post-harvest water use in produce production. And we will have some guest speakers, some of whom were actually listed on your team. So that'll be great. And then I think Faith Kreitzer is going to be one of them. And then April 25th, we've got a guest speaker coming on to talk about just some back to basics post-harvest cleaning and sanitation. So some real hands-on, like this is how to do it. So if you registered for this webinar, you will get the alerts to attend the other two. All of them will be recorded, and my hope is that we can get some Idaho State Department of Agriculture pesticide credits for the last two, because we are going to specifically talk about sanitizers and their use and the use of EPA labeled sanitizers. So that's what's coming up. And then a quick reminder that all of our past years of webinars have been posted on a YouTube that we have, and some of them are on our extension website. I will include that in a follow-up when I post all of the recordings. So if any of these webinars spike your interest, they're all available to watch for free right now online. All right, so that's all that we had for today. I didn't see any other questions pop up. If you've got a burning question, go ahead and type that right away in the chat or the Q&A. When we close this webinar, a brief survey, getting some feedback on the webinar is going to pop up on your screen. We'd really love to get your feedback because it helps us to continue to plan these to meet your needs and to improve them. I want to thank Dr. Waite-Cusic very much for her time this morning and for sharing that really incredible and relevant research. It looks like it would be a lot of fun to be part of your team.
Oh, it is. Crazy, but good. Thank you for having me.
All right. Well, thank you, everyone, and have a great rest of your week.